Look, I actually knew Martin Buerger a little, and his technician, a guy you may have heard of named Charles Supper. So that makes this doubly sweet. Not Bob sweet, doubly sweet. It's also so nice to see so many old friends here. Thank you for coming. That warms my heart. I'm not a terribly sentimental guy. I don't get misty about a lot of things, but this is special to me, and I do appreciate it deeply. I had to think a lot about what to say to you, and before explaining why I chose the topic I did, let me make two comments. One is that many of you may be surprised to see me affiliated with Cornell Medical School in New York City. You would normally associate me with Brandeis University, where my colleague Dagmar Ring and I worked together for so many years. And indeed, until a year ago, I was a professor at Brandeis University, but my wife, Lori Lyncher, became dean of Cornell Medical School in New York City, the first woman to head a major medical school in the United States. And when your wife says to you, "You're going to join the faculty here, right?" You understand what the Godfather, Don Vito Corleone, meant by, "I'm going to make you an offer you can't refuse." But I still do retain an association with Brandeis. Dagmar is still at Brandeis, and we continue our 35-year collaboration, albeit now by using Amtrak. That's the first thing I wanted to say. Second thing I wanted to say was there were two things I could have talked to you about. So one would have been our recent efforts, Dagmar and I have been doing this for about 10, 12 years now, to find treatments for the most severe neurologic disorders. Over that period of time, we've made a little bit of progress. We've identified new targets for 3 of these diseases and have therapeutics marching toward the clinic. I think we will be in human trials with a gene therapy for Lou Gehrig's disease in 18 months if everything goes well. And I'm hoping that in 2 to 3 years, we will be in clinical trials with a drug we have for Parkinson's disease. Alzheimer's is proving a much tougher nut to crack. We have a new target and some prospective chemical matter, but I don't know yet how far that will go. I do know this is what I want to focus on for most of my remaining scientific years. But I wouldn't, I think, choose that topic for this audience because I wanted to do 2 things. One was to sort of wrap up 40 years of working on structural enzymology because it allows me to talk about some of the wonderful people I've worked with. And the other is because I think there is something very important that I want to say to you about crystallography and about structures of proteins. And this is a good opportunity to make that remark, which will come at the end of the lecture, so don't leave. As Martha said, I started out as a graduate student in Oxford with David Phillips, who's seen here with his first student, Louise Johnson. And in 1965, they determined the structure of hen egg white lysozyme. The first enzyme whose structure was determined crystallographically. And 4 years later, I joined their lab, got my PhD, went on to Paris as a postdoc. More about that in a few minutes. It was a wonderful experience. By the way, in the days before molecular graphics, you drew structures by hand. This is the fold of hen egg white lysozyme drawn freehand by Sir Lawrence Bragg, as in Bragg's Law. One of my prized possessions. When I got to Wayne State, it's a medical school, so no undergraduates to work with, very few graduate students in Detroit in those days. And a friend of mine, Tony Fink, about whom more in a moment, said he had an undergraduate at UC Santa Cruz who was so bright, he didn't know what to do with it. So he sent him to me for 6 months. The undergraduate was named Tom Albert, and here he is at age 20, the day I met him. It was a trifle disconcerting. Of course, over time, Tom softened up a bit. Here he is 35 years later, when he was professor at UC Berkeley. Tom was extraordinarily bright and energetic, and so the two of us set to work to try to do something that I wanted to do for some time. It was to trap an intermediate and enzyme catalyzed reaction in the crystal and determine its structure by X-ray crystallography. And we decided the way to do that was to take advantage of a flow cell invented by Marilyn Gosher and Demetrius Cernoglu, and to flow substrate into the crystal at a temperature low enough that the reaction would not take place, and then do the structure. The enzyme we chose was pancreatic elastase. We were trying to trap the acyl enzyme intermediate, which was, this is a serine protease, and the enzyme intermediate is an acyl derivative of the eponymous serine residue. Now, Tom actually succeeded, as I'll show you, in doing that. This is just one of a huge host of firsts that Tom Albert can get credit for over the course of his career, the first three of which he did when he was an undergraduate at UC Santa Cruz in 6 months in my lab. Some of the rest he did when he was my first graduate student at MIT a few years later. You never forget your first graduate student, I think, right? There's something really special about that. Tom was my first graduate student. Anyway, while he was an undergraduate, we did this experiment, and it was based on the idea of Arrhenius behavior. The rate of an enzyme catalyzed reaction diminishes with temperature, roughly a factor of 2 for every 10 degrees if you have a typical activation enthalpy. And we succeeded. We were actually able to do that. This is in the days before graphics, gang. You drew the electron density maps by hand. And here's the electron density of elastase. You can see the solvent boundary there. This is the active site with the various residues marked. And this is a difference map after flowing in a substrate at minus 55 degrees. That's the substrate, and you'll see in a moment when you put them together that this is acylated to the serine residue. It's covalent, you can't wash it out at low temperature. And we used to collect data in those days one reflection at a time by means of diffractometry. Okay? I still find myself sounding a lot like my father. When I was your age, I say to my graduate students, we used to collect reflections one at a time. Okay? It took months to collect data sometimes, and we had to walk 10 miles through the snow to do it. Those of you who haven't quite gotten to my age yet, trust me, you become your parents. But the great thing about collecting one reflection at a time is you can sit on it. And you can use a flow cell to flow substrate into the crystal at low temperature and watch those reflections change. And that tells you not only the kinetics of substrate binding in the crystal, but it tells you when you've reached saturation, and so you can collect the data. And you can see that in about 20 hours, we achieve saturation with this ester substrate. And so here's what I showed you a moment ago, and when you put these maps together, you get, in fact, the substrate acylated to the serine. It's beautiful. And we published that in Nature, Tom's first paper, when he was still an undergraduate. But this is a 3.5 angstrom resolution. And at the temperature we were working at, it needed to be a 3.5 angstrom resolution. We didn't have time to collect higher resolution data before the substrate would turn over. So we then decided we wanted to try to get to 2 angstrom resolution, which meant using a lower temperature. So we dropped the temperature to minus 65 degrees and flowed substrate in. And all we saw was sulfate now. Sulfate is what you see when you do the structure at room temperature. The active site likes to have a sulfate ion. This was sort of upsetting to us. And over a number of years, we and others tried every modification of this strategy we could think of, including changing the viscosity of the liquid and pumping the liquid through the crystals faster. I don't want to tell you the number of things we tried. And always operating at minus 55, minus 60, minus 65, sometimes minus 75. And all we ever saw was this. We couldn't figure out what the heck was going on. Well, We weren't just standing still scientifically during this period, of course. We were collaborating with a large number of people. Some of them I want to mention here. Tony Fink, who really invented cryo-enzymology as a proper field of kinetic study for proteins, especially proteases, was a faculty member for many years at UC Santa Cruz, and one of my best friends who died tragically too young of pancreatic cancer. Hans Frauenfelder, professor of physics at the University of Illinois, now 92 years old, and also one of my best friends, an expert on protein dynamics from a physicist's point of view. Martin Karplus, who better need no introduction to any of you, and who's still an active professor at Harvard, and of course my first collaborator, Demetrius Serenoglu, who's now retired from his post as professor at Rome. And of course, if I'm going to talk about the people I'm working with, one stands out among all others. I can't tell you what the best day of your life is. You know, it might be when your grandson's born, or the day you get married, or who knows what. But I know the best day of my scientific life. The best day of my scientific life was the day Dagmar Ring and I decided to work together. And for the last 35 years, I've had the enormous pleasure of virtually every day of my academic career, knowing that the person in the next office cares about exactly the same things I care about. And if you think you can put a price on that, believe me, you can't. This is someone who should be standing up here giving this lecture one of these days if there's any justice in the world. Finest scientist, nicest person I ever worked with. And she herself has a collection of firsts to her name that are worth noting. By the way, I'm not responsible for any of these, including the invention of fragment-based drug discovery. It's entirely hers. Okay? And yet you'll find that virtually everything I've done of note scientifically, she's been involved in. That should tell you something about the quality of Dagmar Ring as a scientist. Well, one of the things we wanted to do at that time was to see if we could show that proteins in the crystalline state actually exhibited some dynamic behavior. And we elected to use Debye-Waller factors for that purpose. Most people at that time thought Debye-Waller factors were dominated by static disorder. But we reasoned that if that were true, if we did structures at two different temperatures, the Debye-Waller factors shouldn't change very much. Whereas if they were largely reflective of motion, they should get smaller when you lowered the temperature. And sure enough, that's exactly what they do. This is the temperature coefficient of the Debye-Waller factor for different atoms in myoglobin plotted as a function of residue number. And you can see that there's a substantial temperature coefficient for nearly all of the atoms in the protein structure. And indeed, it's very clear that although static disorder exists in crystals, it does not dominate most crystals. The true motions are reflected in the Debye-Waller factors. When I first gave this lecture, by the way, in Sweden, there was an extremely elderly gentleman in the front row, and he proceeded at the end of the lecture to ask a number of extremely acute questions. Okay? Ivar Waller. At that time, 94. We also wondered, and this was actually Dagmar's idea, we also wondered whether as we lowered the temperature, the protein might contract. And if so, we could measure the thermal expansion coefficient of the protein by measuring its volume as a function of temperature. We did this over 9 temperatures, and we published this. And this is a distance diagonal plot. If there were expansion, the points would be down here. As you can see, what there are is massive contraction. And in fact, over a period from room temperature to liquid nitrogen temperature, the volume of myoglobin shrinks by a little over 3%. That's a lot. And we'll come back to that number in a few minutes, by the way. Anyway, that was kind of fun. And we also wanted to look, of course, at the temperature factors, the B factors, as a function of temperature in more detail. And if you plotted them as a histogram, you saw some amazing things. So at room temperature, you got a very broad distribution that was centered at relatively high values. But once you got below about 220 Kelvin, the distribution sharpened into what looks like a Maxwell-Boltzmann distribution, a classical one. and was shifted to much lower values. So there was some kind of transition in the dynamic properties occurring here, and that was also reflected in the overall B factor as a function of temperature. Somewhere around 200 to 220 Kelvin, depending on the rate of cooling, there was something going on. Okay, and you can see that here. It's a broad transition, but it's a real one. And it can be determined by a huge number of techniques, Mossbauer scattering, infrared spectroscopy, you name it, you can get this transition. The trick is the rate of cooling determines where the transition occurs. The slower you cool the protein, the lower the transition temperature shifts. So it's a somewhat fuzzy defined transition. Recently, some people at Cornell Ithaca, where I also have an appointment headed by Saul Gruner, have actually shown that if you use pressure to create amorphous ice in the crystal, you can shift this transition as low as 100 degrees. So it's kind of an interesting phenomenon. And we and Martin Karplus established, I think, pretty carefully that this is mostly due to the behavior of the bound solvent layer and to its change in dynamics as a function of temperature. There are well-defined properties of water going to glassy-like behavior as a function of temperature, and that seems to drive the behavior of the protein in which you're looking at. So we wondered whether this would affect function, and in 1992, led by Ann Stock, who was a postdoc in the lab at the time, now a professor at Rutgers, we showed that if you tried to bind an inhibitor of ribonuclease below this glassy transition temperature, you couldn't get it to bind. But if you tried to bind it above the glass transition temperature, it bound quite nicely. And of course, that got our attention, because the glass transition temperature is right around where we were playing around with elastase more than 10 years earlier. Our early elastase experiments were done above the glass transition temperature at around minus 35, minus 40. That's where we bound the substrate. Later, trying to get to higher resolution, we tried to bind it at minus 55, minus 60. That's below the glass transition temperature. No wonder we couldn't see anything bound. We needed to do the experiment in a completely different way. This, by the way, is Mossbauer data for myoglobin showing the glass transition temperature can also be obtained by other techniques. But the idea is very simple. What we should have done is to try to bind the substrate just above the glass transition temperature and then rapidly drop below the glass transition temperature for data collection. If we did that, it should work. Okay? In other words, we needed to use these 2 points. Well, so we tried this with 2 substrates. The original one that failed, and the new one, a peptide substrate, which would be an even harder bar. And if you do it this way, you bind it at minus 26, you collect data at minus 55, you see a beautiful acyl enzyme now at 2 angstrom resolution. And the same is true of the peptide. Real peptide substrate trapped in elastase. So that explained it. And of course, that immediately meant that the way to look at a lot of complexes of a lot of protein crystals was to flash freeze the crystal after a very quick exposure to substrate. And this is now a routine experiment in many laboratories in protein crystallography. Okay? And that's why it works. It works because you're above the glass transition temperature when you bind, but if you get below it rapidly enough, no further turnover can occur and no product release can occur. And that's why those experiments are feasible. How about that? And by the way, first person to ever flash freeze a protein crystal and do its structure was Dagmar Ringham back in the 1980s. Okay, so during a period of about 20 years, Dagmar and I tried to find a large number of techniques that could be used to trap enzyme-substrate complexes and study them. And we developed these techniques depending on the system. In some cases, you could just look at equilibrium by soaking directly with substrate. That's here, for example, this is mu to rotate, what we did. In other cases, you need to do kinetic trapping. We use that for xylose isomerase. You make a pseudo-steady state with a flow cell. In other cases, you do the flash freezing. I just showed you that. And then, in some cases, you needed to use very rapid data collection and maybe initiate the reaction by something like photolysis of a cage substrate. I'll show you that in a moment. Well, the flow cell technology we still use. We really like it. I'm surprised more people don't use it. It's not that hard to build a flow cell. And it's an easy thing to use if you want to flow substrate into a crystal. And if you do that constantly, you'll set up a pseudo-steady state, and what you'll observe when you collect the data is the species prior to the rate-determining step. And there's no better way to get it. It's a beautiful way. And a number of labs have used this, but more on to it. That, by the way, is a Cytochrome P450 crystal. That's why it's red. And then, of course, there's rapid data collection by the Lowry method. That is a technology that you absolutely need if you need to collect rapid data. This is the days before free electron lasers, but nevertheless, it's a great way to get data on a very rapid time scale. But you better have a crystal that's really well ordered, because otherwise you're just going to get very smeary diffraction. We first used the Lowry technique combined with flow cell and photolysis to determine the structure of the GTP-bound form of the cancer-causing protein RAS. And this was the PhD thesis of Ilma Schlichting, who was then a graduate student at Heidelberg, where I was on sabbatical, working with my graduate student, Steve Almo. And the two of them together got this experiment to work. We published it in Nature, and here's the GTP-bound form. That was the first structure to show the conformational changes involved in going from GTP to GDP of this protein, and it ended up getting the Max Planck Prize for Roger Goody and some clown from the United States. Okay, so now we have this arsenal of techniques that can be used. We decided, when Ilma joined our lab as a postdoc, that we would take advantage of that and try to do the structure of every kinetically significant intermediate in the reaction cycle of an enzyme. To do this, we needed a lot of things. We needed a tractable system. And after some consideration, we decided that our best bet was Cytochrome P450, which in the immortal words of George Kenyon, is nature's equivalent of a blowtorch. Because Cytochrome P450 can take unactivated oxygen and insert it into a carbon-hydrogen bond, and it can do that at room temperature, whereas normally you need a blowtorch to do it. And we thought it would be wonderful to look at the kinetic intermediates of this extremely complex and fascinating enzyme. So we collaborated with Steve Sligar, an expert on the enzyme, to do this, but we also needed Bob Sweet, who happens to be here in the audience, because without Bob, we wouldn't have access to the synchrotron beam we needed to do these experiments. Now, Bob, as I'm looking up this stuff to put this talk together, I realized that we worked on this with you over a period of about 10 years. And Ilma found like 8 different crystal forms of P450, all of which we then had to schlep out to Brookhaven and try out. Of course, they didn't work, so we sent to schlep them back. And as I recall, we never actually formally applied for time. And we would basically call up because Bob and I go way back. We go back actually to the UK, okay? And so I would basically pick up the phone. I would call Bob. I would say, Bob, we need time. Otherwise, I'm going to tell people about the following things that you did when you were a graduate student. And, this worked beautifully. Have I ever thanked you for that? Well, I'm not going to, so don't worry. All right. So this was great. I mean, literally, without Bob, who, also, by the way, made all kinds of modifications to synchrotron beam station to accommodate us, without Bob, this would never have happened. And, as I said, an immensely complicated enzyme with immensely complicated problems. I'm only going to mention one of them. Okay? This is a 2-electron reaction, and the 2 electrons come in separately. The first electron can come from anywhere, so we use dithionite, no problem. The second electron can only come from another protein, cytochrome P450 reductase, which did not happen to be in the crystal. And so we had the colossal problem of figuring out how to inject the second electron into the reaction. I'll tell you how we solved that in a second. This is Ilma, now a postdoc in the lab, okay, at that time. This is Kelvin Chu, this is Joel Berenson, this is the National Synchrotron Light Source, and somewhere in there Bob Sweet's asleep in his office. Well, it took 10 years. 5 of which Ilma spent as a postdoc in the lab, 5 of which she spent continuing to work with us on the problem when she was an independent scientist, namely the second woman ever to direct the Max Planck Institute in Germany. And we eventually got it to work. This is the dioxygen complex, which has never been seen before. This is the heme group, flat here, as you can see it. There's the dioxygen, bent, attached to the iron atom in the center of the heme. And these water molecules turned out to be crucial for the chemistry of the reaction. Nobody knew the source of the protons for this reaction. We were able to show that they were one of these water molecules by mutating the residue that held it in place, and that's another story for another day. This is the camphor substrate. Now to get to the activated oxygen intermediate, where this oxygen is split off, and you're left with a single oxygen of unknown geometry, which everybody's been arguing about for decades. You had to inject the second electron. And this was probably the best idea Dagmar and I ever had. Because we realized there was one other source in the universe of electrons hot enough to reduce this protein. The X-ray of a synchrotron. Because when you radialize water using high wavelength X-rays, you produce, among other things, hydrated electrons, and they're hot as a piston. So what we did was to blast the protein with electrons at above 1.5 angstrom wavelength, and then rapidly drop below 1 angstrom wavelength for data collection, where the concentration of hydrated electrons was much lower, so we did no further electron damage to the crystal. And this got the activated oxygen intermediate, which turned out to be an oxoferrol species where the iron-oxygen distance was 1.65 angstroms. And that's the first time anything like this had ever been seen before. And this has held up remarkably well, both in other crystallographic studies since the year 2000 when this was published, and in a huge number of spectroscopic studies that have also employed the trick of using electrons donated by the X-ray beam itself. And this was what we published, the movie of Cytochrome P450, the structures of every kinetically significant step in the reaction catalyzed by an enzyme. I'm still enormously proud of this work, which required the efforts of one of the finest postdocs I ever saw, Ilma Schlichter. And I've had some great ones, Steve Burley, Axel Brunner. Ilma did unbelievably great work on this problem. Okay, now the issue of how proteins function is one that has interested us since the 1970s. And in 1983, while a graduate student in my lab at MIT, Tom Alber and Dagmar Ringe, together with postdoc Bill Gilbert, came up with a hypothesis. The hypothesis was called the directed fluctuations hypothesis. It's now known as promoting vibrations. The idea is that at a given temperature, okay, things are moving around in the active site. If they're moving randomly in all different directions, that's one thing. But what if the geometry of the active site constrains the motions so that they lie preferentially along the reaction coordinate? That might facilitate the crossing of the transition state better. In 1983, we could only propose this. We couldn't do much about it. But in the last 15 years or so, this has turned out to be a pretty hot topic in structural entomology, and people like Steve Benkovic and Peter Wright and Charlie Brooks and Sharon Hamish-Schiffer have done some wonderful work on enzymes like dihydrofolate reductase, showing that this is very likely to be the case for at least some enzymes. Well, at the same time, we were curious about a paradox that had first been pointed out by Tom Brock the discoverer of thermophilic enzymes in the United States. They were simultaneously discovered by Taro Oshima in Japan. Tom Brock noted that the catalytic activity of thermophilic enzymes at ordinary temperatures is almost zero. And I can assure you that's true. Here's an example, okay? There's the thermophilic enzyme, which has optimum activity about 80 degrees, and you can see that at ordinary temperatures, its catalytic activity is zilch. Well, you know, what's causing that? Brock had no explanation. We thought we might have one. The idea was that maybe in order to become thermophilic, you had to make a pretty rigid structure. And maybe it was too rigid at ordinary temperatures to have the kind of promoting vibrations and other things that you need to catalyze a reaction. And so, working with Peter Zawadzki, who's now Vice President of the Hungarian Academy of Sciences, and Taro Oshima, a wonderful friend in Japan, graduate student Gerlin Wallen, who now runs the postdoc program at EMBO, by the way, Gerlin then took a look at this. And here's hydrogen-deuterium exchange measurements of a bunch of isopropyl malate dehydrogenases. And what you can see is that as you go from an enzyme whose temperature optimum is in the 20s, to an enzyme whose temperature optimum is in the 30s, to an enzyme whose temperature optimum is in the 60s, the enzyme becomes progressively more rigid. The hydrogen-deuterium exchange rates become slower and slower and slower. But if you look, and this is at 25 degrees, but if you look at these enzymes and do the hydrogen-deuterium exchange at their optimum activity temperatures, they're all the same. They're all equally flexible at the temperatures at which they are optimal. I really like this experiment, and we went on to see if that was reflected in the temperature factors, and of course it was. The temperature factors for the thermophilic enzyme had much less variation than the temperature factors for the mesophilic enzyme. So all of that suggested that there's something to this notion that you really need intrinsic flexibility in order to get not just binding, like we'd shown earlier, but really to get catalysis. And so Mark Wilson, who came out of Axel Brunner's lab and then postdoc with us, so our scientific grandson, as it were, Mark Wilson decided to look at dihydrofolate reductase. So a lot of work's been done on dihydrofolate reductase. It transfers a hydride ion from NADPH to tetrahydrofolate, to folate, I should say, and here you end up with tetrahydrofolate, and here's the active site. The hydride transfer goes like this. And there's a lot of interest in this loop containing methionine 20 because Charlie Brooks, with some molecular dynamics calculations, and Peter Wright, with some NMR calculations, had all shown that that loop was flexible and should be very flexible in order for catalysis to occur. But in the crystal structures of dihydrofolate reductase, there was really no evidence for that flexibility. So we looked at the structure at 4 degrees Celsius, not at 80 Kelvin, but at 4 degrees Celsius. And we not only saw interesting vibrations of the substrate and cofactor where the atoms were driven towards each other, but we saw this enormous vibrations of the methionine 20 side chain, okay, in terms of their anisotropic thermal factors. This structure was done with below 1 angstrom resolution. So here you have a situation where there's a rich variety of motions that are only revealed when you look at the structure at 4 degrees. The existing structures we'd had up to that point had all been done at 80 Kelvin. And they just showed nice little circles around there. Well, right around that time, Tom Albert, professor at Berkeley, and his student, Jamie Fraser, started to wonder independently, very independently, whether there weren't problems with low temperature crystal structures. And in a paper they wrote in 2011, they found that more than 35% of side chains were remodeled as you went from room temperature to low temperature in protein, and packing defects were completely altered. Not surprising when you remember the thermal expansion coefficient, and that this protein is shrinking as you lower the temperature, okay? They said this result exposed a bias in structural databases towards smaller, overpacked, and unrealistically weak models. And here are some examples of what they saw. This is all published, so I'm not going to go through it in detail. They found for a number of proteins where they were able to do both room temperature and low temperature structures, they found an average thermal expansion from room temperature to 80 Kelvin of 3.4%. Remember, we found 3% or so for iodines. This was very gratifying. And you can see the effects on the cavities. The cavities with numbers in them are cavities that differ between the room temperature and low temperature structures. And there's also substantial changes in side chain positions as you go from room temperature to low temperature. This would be interesting by itself, but in this case, these being active site residues, this is more than interesting, this is troubling. Now, we were looking at dihydrofolate reductase. Remember, at very high resolution. And one of the things, of course, we saw was that dihydrofolate reductase at very high resolution occasionally allowed us to determine the positions of protons in the electron density map. You see, we were troubled by the low temperature business. We were worrying about whether or not the well-known fact that active site residues have perturbed pKa's might mean that if the pKa's were temperature dependent, then maybe in an 80 Kelvin structure, you really have no idea what the protonation state of your active site residues would be. And indeed, Pierre Duzou, in whose lab I did my postdoc back in the 1970s, had shown, this is our work together back then actually, that many different chemical groups have pKa's that are sharply temperature dependent. Look at Tris. Okay? This is over a range from 20 degrees to minus 60. May I point out to you, cryostructures are typically done minus 200. Just extrapolate some of these curves, and you'll see that you can get pKa changes of 2, 3, 4 units, okay, over a range from room temperature to minus 80, depending on the chemical group. This is a big worry. Okay. So Louis Lazar, shown here in his wedding picture, and Cheryl Kreinbring in our lab decided to look at this, and to do that, they took protein, gamma-chymotrypsin, and they did 12 data sets. The data sets were all done at around 1 angstrom resolution. The data sets were, in some cases, room temperature, in some cases liquid nitrogen temperature, and they were done at 3 different pHs to look for protons in the electron density maps. But of course, if you're going to do that, you need a gold standard for whether the protons are really there. So at the same time, we also did neutron diffraction at room temperature at the 3 different pHs. So as you can imagine, we're still working some of this stuff up. But it's kind of interesting. We found that you could easily tell the difference between a protonated and an unprotonated aspartate or glutamate from the heavy atom distances alone. If you had enough resolution, and in this case 1.1 was plenty, okay, your precision and distances were enough that you could tell whether you had a proton there, whether you could see the electron density or not. Okay. Similarly, you could tell the protonation states of histidines and tyrosines and so forth. Now, that's really very encouraging. But it's also troubling. You see, right around this time, some NMR work came out on alpha-lytic protease, another serine protease, which showed that the pKa of the aspartic acid in the catalytic triad was 2, which means that at pH 5 it should be unprotonated. And indeed, in our room temperature structures, from the distances, it was clearly unprotonated. In our cryo structures, it was protonated. Exactly as the temperature dependence of the pKa would predict it should be. How many other proteins in the data bank have this problem? Okay. Well, we're still working on the neutron structures, but this is, of course, a really important role for neutron diffraction to provide us with some gold standard measurements so that we can then figure out how to best use X-ray data, which are easier to get, to really determine protonation states. This is some of the neutron work which is still in progress. Well, last year, we started collaborating with Jamie Fraser, who'd left Tom's lab and now had his own lab at UCSF, to look at dihydrofolate reductase again. And Mark Wilson had now left our lab and was professor at the University of Nebraska, so the two of them and Dagmar and me worked together on this problem. Nice to work with your scientific children and grandchildren, actually. And we published this, so I'm not going to go through it in detail, except to say it was pretty terrifying. When we looked at two different room temperature data sets, compared 8 years apart on dihydrofolate reductase collected by different labs, we saw they were incredibly comparable. We looked at two different 80 Kelvin structures of dihydrofolate reductase collected by the same two labs 8 years apart. There were enormous differences between them. And this was caused in part by slightly different cryoprotective protocols, different rates of cooling, slightly different packing in the unit cell due to the rates of cooling, and so forth. If this doesn't scare you, it should. It scared us. That was last year. So where are we now? Well, where we are now is that during the last 25 to 30 years, and I'm partly responsible for this, okay, nearly all the structures of the protein data bank have been determined at low temperatures, very low temperatures, more than 100 degrees lower than the last transition in protein dynamics. And we know that those temperatures have clear-cut effects on dynamics and the volume of the protein and the packing of the protein. We also know they have unpredictable effects on side chain conformations, domain positions, protonation states of residues, and metal ion spin states, which, by the way, are really temperature dependent, among other things. Which means, basically, that most of the structures in the protein data bank are of unknown state. We don't know whether they represent an active conformation or not. Probably they're fine. Every time I talk about this to people, they say, well, you know, it seems to me they're probably fine. Yeah, they're probably fine. But I'd like to know for sure. And I think you ought to know for sure, too. So here's my proposal. I think we should make a concerted effort to determine at least one room temperature structure at the highest possible resolution for every unique protein and protein data. That's a lot of work. Consider it job security. But you know what? It might not be as much work as you think. I mean, we used to do this all the time. We used to take 20, 40 crystals per data set. Bob, what was the most crystals you ever used for a data set? In my case, it was like 30. I don't know. Yeah, there's lots, right? A ton of crystals. And Bob Frederick, I think, had to use one crystal per frame for phosphorylase, as I recall. So, you know, I mean, we used to do this. We scale it in, and it was okay. All right? But you know what? We might not have to do that. Because I think this could be one of the best possible uses imaginable for free electron laser crystallography, which can be done at room temperature really easily and can be done with small crystals really quickly. And while we're waiting for single particle and other uses of free electron laser crystallography, this might be a great use for the community to put it to. We should do this. Well, I was going to end with this because 5 years ago, we had a party in the lab to celebrate 30 years of Dagmar Rena and me doing structural biology. And, you know, that was going to be the last slide. To talk about how wonderful it's been to work with her and all the other people, some of whom I've talked about, some haven't had time to talk about all the others. I'm sorry about that. I had to make some choices about just restricting the material. But I've been so damn lucky, and every one of them, okay, everybody's ever gone through the lab has contributed in some way or other to my standing here and giving this lecture. But when I looked at this picture, I had a great Gatsby moment. Because see, over there is Tom Albert. The last line of The Great Gatsby is, "So we beat on boats against the current, borne back ceaselessly into the past." And this drew me back into the past. To that year when Tom first came into my lab. Here he is on the Piper Trail in New Hampshire. First mountain we ever climbed together. Tom died when I met many years later. And here he is in 2010 on the high Sierras. He was a great mountain climber, loved to climb mountains. The picture was taken in 2010. 4 years later, Tom Albert was dead of Lou Gehrig's disease. He died last year at the age of 60. When somebody young like that dies, I mean, I don't know, maybe it means something, but the older I get, the more convinced I am that I don't know what anything means. In fact, that I don't know anything at all. But I do know this. I know that if Tom Albert were still alive, I'd take that award, wonderful as it is, and I'd put it in his hand. Because Tom Albert was my first graduate student. He believed in me when there was nothing to believe in. And I know this too, that Tom Albert is and always will be on top of a mountain higher than any I will ever climb. Thank you very much.